My name is Arshia, and I'll be talking about the discovery, synthesis, and optimization of covalent inhibitors that target the G12C mutation in KRAS, which can cause cancer. So, sorry. A brief outline. First, I'll be talking about how KRAS was originally considered to be an undruggable target. Then I'll talk about the progression of the G12C covalent inhibitors, and then on to the pharmacophore model that we created, starting with the southern hemisphere pyrazine, and then the isoxazole on the northern hemisphere. And then I'll talk about the analog library synthesis, optimization, structure-activity relationship, and some preliminary biological activity. And we'll conclude with some future directions and acknowledgements. So KRAS is a protein that is part of a signal transduction pathway that regulates cell growth and proliferation. And mutations in KRAS can cause up to 25% of cancers. The most common mutation site on KRAS is on the 12th codon, and the G12C mutation specifically makes up about 14% of those mutations. The G12C mutation is most common in non-small cell lung cancers. It makes up about 12% of those cancers, and it also accounts for about 3% of colorectal cancers and 1% of pancreatic ductal adenocarcinomas. So the way that the mutation works is the GTP-GDP cycle is continuously locked in the active state with the GTP bound to the switch 1 domain. And this causes uncontrollable cell proliferation, which leads to cancer eventually. And originally, KRAS was thought to be undruggable because of the picomolar affinity to GTP that keeps it locked in the active state, and the featureless, nearly spherical surface of the protein, which makes it very difficult for any compounds to competitively inhibit at that particular site. However, through the usage of covalent inhibition, we can actually inhibit this mutation. Covalent inhibition is found in several FDA-approved drugs, and they all use a warhead, which is highlighted in purple. And there are many different covalent warheads that can be used. Some of them are shown here. And the one that we were originally focusing on is the acrylate warhead, which targets the cis-thiol and engages it in an irreversible Michael addition. So the inhibitor can irreversibly bind to that particular cysteine 12 residue, which is what we're targeting in the G12C mutation. However, we've also recently started looking to some other warheads like the fluoroacrylate warhead. And then some of the early inhibitors of the G12C mutation started in Showcat's lab, which their most successful one was inhibitor 12. And then moving on to AMG 510 and MRTX 849. AMG 510 was approved in 2021, but was recently recalled from the market. And MRTX 849 was approved in December of 2022. And below there is the AMG 510 interacting in the KRAS switch 1 domain, and it is binding to the yellow cysteine 12 residue. And then some more recent compounds, which are promising but haven't been approved yet. So there's Genentech's GDC 6036 and Novartis's JDQ 443. And then you may be wondering, well, what do all these compounds have in common? Well, they all have this piperazole fragment highlighted in purple and a warhead in blue. If you'll notice, most of these have the acrylate warhead, but the MRTX 849 actually has that fluoroacrylate. So that's something that we want to probe further. And then using these similarities, we were able to devise a pharmacophore model, which consists of a 2-substituted acrylic warhead that targets the cysteine 12 residue. And then there's the pyrazine spacer domain, the hydrogen bond acceptor, and the aryl fragment. And then 2 of these compounds actually have a S-methyl on their pyrazines. And we wanted to sort of explore if this S-methyl affects how the methyl actually conforms in the pocket and if that makes them better targets for G12C mutation. So we used DFT analysis and compared the equatorial and axial positions with the S-methyl, and noticed that the equatorial position was actually favored in a ratio by a 2.297 kcal per mole difference. However, when we actually look at that S-methyl in AMG 510 binding in the pocket, it's actually pulled out into the axial position instead of the equatorial position. There were a couple hypotheses for why this may be. One was that when the warhead binds with the cysteine 12 residue, it can cause some sort of chain reaction that pulls that methyl on the piperazine out into the axial position. However, when we looked at another compound that has an S-methyl but doesn't actually target the cysteine 12 residue, instead it targets G12D, that methyl was still pulled out into the axial position. So since they're both in the same pocket, we considered the possibility that there is a very hydrophobic pocket in the protein that is attracting that methyl and overwhelming the 2.297 kcal per mole difference. And then in the northern hemisphere of our pharmacophore model is the 5-methyl isoxazole. And the 5-methyl isoxazole is used in several different compounds in medicinal chemistry. There's nucleosin and decloxacillin, and they all have various effects. And so it's not just for anti-cancer drugs, it can be used for antibacterial or antiviral drugs. And the reason why it's so common is because of the oxygen and the nitrogen, which establish strong hydrogen bonding, which help with any hydrophilic interactions in a pocket. And then moving on to our analog library. So the original library consisted of just the 4 carboxylic acids and the 3 piperazines, and just the singular warhead, which was the acrylamide warhead, so W1. But recently we expanded it to include 2 more warheads, so the W2 and W3, which is the fluoroacrylate and trifluoromethyl acrylate. And then there's the whole library drawn out below, so those are 36 compounds now, and but all of them contain those key aspects that I talked about earlier: the warhead, the piperazine spacer domain, the hydrogen bond acceptor, and the aerofragment. And then the synthesis is currently starting with the Bach protection or amide coupling, which uses DMAP, triethylamine, HBTU, and it takes about 18 hours. And then a D protection using TFA and DCM, and then followed by an acrylation using triethylamine and THF. So one of the goals of our project was actually to telescope the reaction and optimize the first step, which was the amide coupling. So we used high throughput LCMS screening and tested out the 6 salts shown as different coupling agents, as well as DMAP and HOBT as the catalysts in 2 different equivalences each. So we did 1 and 3 of each. And then we tested them all out on small scales for 19 hours. And from that, we were able to see that the conditions of 2 equivalences of HBTU and 3 of DMAP gave us our highest yield of 93%. And when we scaled that up, it was about 98.87%. And this is compared to the original conditions, which were with 2 equivalences of EDC and 3 of HOBT, which through this study gave us about 71% yield. So that's about a 20% yield increase. And then here we have the traces of the LCMS of our starting material in pink and the product in blue. And so we basically looked for the product peak and back calculated area under the curve on all our reaction vials to determine the actual crude yield of all those reactions. So we also tested these optimized conditions on all the W1 analogs, or well, all the box, which would eventually become those particular analogs. So one thing we noticed is that amongst the A2 analogs, which was A2P1 was the compound that we actually optimized, so those all have the same carboxylic acid. They all have relatively high yields of 99 and 98%. However, amongst some of the A4 analogs, A4P2 and A4P3 specifically, the yield is significantly lower. One thing we consider is that the fluorine is so electronegative that it's making it very difficult for the acid to activate and then couple with the HBTU and actually form the product. And then we moved on to MTTs. So we have here just sort of a flow chart of what we were doing. So we first tested these compounds against HCB54, which has the G12C mutation, and it's a non-small cell lung cancer. And then we also tested them against 2 cell lines that don't have the G12C mutation, HCT116 and HT29, which are both colon cancers. And then we started with A2P1, A2P2, and A2P3, so all those A2 analogs. All these are W1 because we hadn't expanded our library until recently. And one thing we noticed is that the most effective against these HUB54 cells was actually A2P3, which, as you'll notice, has the R-methyl and not the S-methyl, which we thought was a little interesting given that AMG 510 as well as I believe GDC 6036 both had that S-methyl. And then the IC50 value we gathered from that was 10.45 micromolar. And then we also tested them against the HCT116 cell lines. And for this cell line, we tested all our P2 analogs. So A1P2, A2P2, A3P2, and A4P2. And the most effective was A2P2, or the most selective was A2P2 because it wasn't effective against this cell line, but it was against the actual HCB54, which has the mutation. And then we looked at it against HCT116 cells, and a similar trend was seen. A2P2 was the most selective as it did not actually inhibit the proliferative activity of these cells, while it did with HCB54. And the same goes for this test as well. And then in conclusion, the optimization results were shown to be successful for all the A2 analogs. However, we want to consider doing one with the A4P1 analog because that particular carboxylic acid with the fluorine and the chlorine did not show as good yields with those optimized results. So we were hoping to see if different conditions would maybe have different higher yields. And then another thing we wanted to do was test different fluoroacrylate warheads using 19F NMR and running kinetics on the coupling or the acrylation between the piperazine and the warhead. And then we also want to continue MTT assays on a couple different cell lines like MDA 231 or MCF7 and analyze how some of our compounds are showing anti-cancer activity against cell lines that don't contain the G12C mutations. We want to finish MTTs on the other 24 compounds with the different warheads and do some more in vitro tests and DFT analysis to understand the correlation between the phenyl substituents and the potency. So thank you to ASDRP, our collaborators, Dr. Tracy Zhang's group, and the other KRAS lab members, and special thanks to Dr. New. Any questions?